Hi guys, we're going to be talking about next generation sequencing or high throughput, massively parallel sequencing. And this is not going to have a ton of details about each of the different techniques. This is to supplement sort of what the videos that are already posted on D2L. So let's get started here. At the top you can see we've got a big chunk of DNA, and you can think of this not as like a PCR product, but as something bigger, longer, like a chromosome or 23 pairs of chromosomes. And this DNA has an unknown sequence. We don't know what the sequence of that, I mean, we might know some of the genes, but the big picture story is really unknown to us. So in order to start sequencing this huge pool of DNA, I first want to start by fragmenting the DNA into smaller pieces. And you can do this a couple of ways. You can use sonication, you can use enzymes to cut the DNA. No matter what, we're going to fragment the DNA into small pieces. Depending on the company that's doing the sequencing or the type of chemistry you'll be using, the fragments might be different sizes. But let's say that our fragments are between 150 and 500 base pairs in length. Remember, this DNA still has an unknown sequence, so it would be really hard to use a primer to start the sequencing reaction. And that's where this next step comes in. We're going to add in adapters. Adapters are oligonucleotides, that is, they're short pieces of DNA, and they're ligated, or we use the enzyme ligase to build a phosphodiester bond to attach them to the ends of our DNA sequence. Here they're just shown on the left, but we could add adapters to the right side of each of the DNA fragments as well. What this means, this addition of adapters means that even though we've got here 3 different sequences, it doesn't matter what the green regions are. We can use the same primer that will bind to the adapter for any number of fragments, and we'll be able to start the chain elongation, the use of DNA polymerase to elongate the growing DNA chain. So these adapters help us make our unknown fragments a little more uniform. Once we have our adapter ligated fragments, we're going to capture each fragment on a bead. I'm going to show you, or I just want to remind you that we've got more than one fragment here, and each fragment is going to get its own bead. If we zoom in on these beads, you can see that now we're going to use the adapters with primers that can anneal to them to extend the strand and ultimately make many copies of each fragment. So I'm showing you this one step at a time here, but we're doing this times thousands or even millions of fragments. Now here in the center we're looking at a bead. It's also common for the fragment of DNA to be immobilized or stuck onto a slide for sequencing. And then using this clonal amplification, that's just PCR, we're going to make many copies of the DNA fragment on the slide or on the bead, and then ultimately we can use this for sequencing. The strategy on the right here is often used by Illumina sequencing, and they call this approach bridge amplification. If we look at this image here, you can think of this as the surface of the sequencing slide or plate. And when we're thinking about beads, we'd have one bead in each of these locations. Let's see if I can draw it here. We'll have one bead in each of these hexagonal wells, and each bead will contain a different DNA fragment with many hundreds of copies of the same thing on it. Then using a camera, I don't think I can draw a camera, using a camera, we can watch the entire plate and take pictures of when different bases are added. You can see we've added a base in these wells that are releasing a color here. Let's talk about what that looks like a little more closely. So in the upper left here, we're looking again at our unknown DNA sequence. And I'd like to start by putting in the adapter sequence that all of the DNA fragments have in common. We're going to start by using a primer that will anneal to that adapter, and then the newly synthesized strand will build off of the primer. Well, how do we synthesize that new strand? We'll do that by rinsing a pool of deoxy ATPs over the surface of this plate filled with DNA fragments. And if there is an A that's added next in the sequence, we'll add the DAMP, we'll add the single phosphate onto the chain, and then pyrophosphate will be released. And in the image below, that pyrophosphate release you can see as the green or yellow flash of light. And that's picked up by the camera watching the surface of the plate. So you can see here, this flash of light indicates an A was added in well number 1. We can repeat this again. Let's rinse off the A's, we'll add some C's. And if there's no flash of light, then that means there's no C in that reaction. We've got no C, we've got no G added, but when we rinse off the G's and wash over some T's, we actually have a double burst of light indicating there are 2 T's present next in the DNA sequence. It's really nice that we can, because we're dealing with chemistry here, the amount of light, the amount of pyrophosphate is proportional to the number of bases that are added. Now, this approach that I've just described is called pyrosequencing because it uses these pyrophosphates to show when each base has been added. And a company that uses this technique is called 454. So 454 sequencing is the same thing as pyrosequencing. There are, of course, other ways to quantify the bases which have been added to the DNA. Another approach would be ion torrent. They rinse one base over at a time like this, but in the case of ion torrent sequencing, they look for protons rather than pyrophosphates. Illumina sequencing uses differently colored bases. So A has a different color from T, from G, from C. So they're colored bases. And rather than looking for one flash of light, you're looking for a specific colored flash of light. Now let's talk about the big picture of this, what we do with those many flashes of light. Remember that we started with chromosomal DNA, but we don't know the sequence of it. Then we can amplify using PCR to get many copies of what we'd like to sequence. Sometimes fragmenting comes first and then amplification. Sometimes amplifying comes first and then fragmentation. But either way, we've now fragmented the DNA and we've added those adapters, which aren't really shown in this picture, but you'll recall that from the earlier slide. We've just talked about sequencing the fragments, and we're starting the sequencing using a primer that anneals to the adapter. And the type of sequencing that we've talked about here today is sequencing by synthesis. That is, we're building the second strand of DNA in order to figure out which bases are being added. We're sequencing by synthesizing the second strand, starting with that primer and using DNA polymerase. Once we have used a camera to watch all of this sequencing by synthesis. The camera will have picked up the flashes of light, which mean bases have been added. Using computational tools, we can turn that into a list of reads. That is, short fragments of DNA. It's wrong to call them DNA fragments. These are sequence fragments that came from the DNA that was just sequenced. Now, we get those sequence reads in a random order. And in order to make sense of them, we need to build contigs. That is, we need to look for regions of overlapping sequence so that we can line up many bases together, or many reads together, to build shorter and then longer and longer sequence reads. Ultimately, to build back the chromosome, whose sequence we didn't know at the beginning, but after all of this hard work, we can reassemble that chromosomal sequence. Since our goal is to rebuild this entire chromosome, whose sequence we don't know, we need to build those contigs of overlapping read information to tell us which sequences are next to one another. And that can be challenging, especially if you don't have a comparator, something to compare with. Often this is called a reference genome. So perhaps one cow has been sequenced, but now you would like to sequence 3 new varieties of dairy cattle. You could use the reference genome from the original cow as a reference to build your new genome. What if you're sequencing the genome of a mushroom that has never been sequenced before? There is no reference genome, and we would call that de novo genome assembly because we're not using a reference. And when doing something like that, it can be really useful to use paired ends sequencing. So earlier I told you that our fragments would be about 150 to 500 base pairs in length, because that's about as long as the sequencing reads we can get from next generation sequencing. Now, in paired end sequencing, it's also sometimes called mate pair sequencing, we can use fragments that are much larger than that, maybe 3,000 base pairs in length. So I'll draw a 3,000 base pair fragment. Here's our 3,000 base pairs. Now, next generation sequencing is still only going to find the sequence information for the 150 base pairs on either end. But because we have paired end sequenced them, we're going to know that this fragment of DNA and this fragment of DNA must be 3,000 base pairs apart. Because we're randomly fragmenting the genome, we're going to end up with, let's see if I can draw this. We're going to end up with this piece of DNA and this one that are 3,000 base pairs apart. We don't necessarily know what's in between them. We're going to end up with another piece of DNA and another piece of DNA that's 3,000 base pairs apart, and we don't necessarily know what's in between them. But we can use our other small read fragments to build up related reads nearby and discover that some of the reads that overlap here are also so we can build up a long contiguous or continuous sequence by using these paired end reads to sort of be a backbone, be a scaffold for all of the other reads. And so I'm drawing these little reads here these little bars here to indicate that I have another short 150 base pair fragment that overlaps some sequence with this region. And so I'm starting to build my contigs by piling up those reads where the sequences match. This can also be really useful as we're trying to fine map a genome. So fine mapping, where we're trying to really clarify all of the different parts of it. And some of the hardest parts to clarify can be the centromere and the telomeres. That is the middle of the chromosome and the ends, the tips of the chromosomes. And that's because these are full of repetitive DNA. They're really repetitive, which means it's hard to know if we have many reads of the same sequence. We just have, 400 of those reads. Or do we have 400 copies? Or is this 400 bases long? It's hard to know. Have we sequenced it many times, or is it really long? We can't know if it's really repetitive. And so by using paired end sequencing like we've drawn up here, that will allow us to put some spacing or determine almost the length of the repetitive region. You can do this, of course, with DNA that's not repetitive, but when it's really hard to build these overlapping contigs, the paired end sequencing can really spread out the context of the repetitive DNA that you're interested in. So that's one of the benefits of using paired end sequencing. The very last thing I'd like us to look at is an assembled contig. So ultimately what we want to do is build back a chromosome sequence from these many fragments of sequence reads. So this is fragment number 1, and this is fragment number 2, and fragment number 3. They were all sequenced using one of those sequencing by synthesis methods. And then computationally, using algorithms, we're going to line up these sequences where they match. So maybe we've got TAATCG here, and in this sequence we've got ATCGAA here. Looks like this sequence is going to be ATCG AATATT, and then maybe do all the way up here we've got ATTCCGAC. Okay, this one is also TTCCG, and that one's shorter actually. So we're using the sequence similarity to be able to line up all these fragments, and ultimately we'll be able to assemble the entire sequence of the chromosome. TAA, TCG, AA, TATT, CCGAC, and we can build the entire chromosome in this fashion. So this is one of the things that next generation sequencing helps us do. It's to build the sequence of the chromosomes so we can know exactly which genes are located exactly where on each chromosome. Now, what do those genes do is a whole nother story.